Welcome, everyone, to our deep dive into the world of oral cell examination. This isn't just a simple science experiment. It is a powerful technique that lets us study our own epithelial cells and even detect diseases at their earliest stages. Today, we're going to cover everything you need to know. We'll explore the different types of cells you can find in your mouth, how to properly collect a sample, the secrets behind preparing a slide for the microscope, and what to look for when you're observing the results. Plus, we'll answer some of the most common questions people have about this fascinating process. So grab your lab coat, and let's get started. First, why look at oral cells? They're incredibly easy to access, and they regenerate very quickly, which makes them perfect for both research and diagnostics. Inside your mouth, you'll mainly find a few key types. The most common are superficial cells. These are mature, large cells with tiny, condensed nuclei. Then we have intermediate cells, which are a bit less mature. You can spot them by their clear, round nuclei. Basal cells are much rarer to find in a simple swab. If you see them, it might be a sign of some kind of tissue damage or disease. Finally, you might see inflammatory cells, like neutrophils and lymphocytes. Their presence usually tells you there's an infection or inflammation happening. So, what's the main goal of examining these cells? It's a non-invasive way to check the health of our oral tissues. This simple swab can be the first step in detecting some serious conditions. For example, it's used for the early detection of oral cancer, helping catch it before it becomes more dangerous. It can also identify viral infections, like HSV, the herpes simplex virus, or HPV, the human papillomavirus. Beyond that, it helps us spot precancerous cellular changes, known as dysplasia. We can even see the effects of environmental factors, like how smoking or a poor diet can change our cells at a microscopic level. It's amazing what a small sample can reveal. Now for the hands-on part, the experiment itself. To do this, you'll need a few things: a sterile cotton swab or a brush, some clean glass slides, a fixative like 95% alcohol, and Papa-Nicolau stain, which we'll talk about more in a minute. The collection process is straightforward but needs to be done carefully. First, rinse your mouth with water to clear away any food, debris, or loose cells. Then, take your sterile swab or brush and gently but firmly scrape the inside of your cheek or the side of your tongue. You want to collect a good layer of cells. Immediately after scraping, you'll transfer the cells directly onto a clean glass slide by rolling the swab across the surface. The most critical step comes next. You must immediately fix the sample with the alcohol. This preserves the cell structure and prevents them from drying out or distorting. Without proper fixation, your results won't be reliable. Once the sample is collected and fixed, it's time for the magic of staining. We use a method called the Papa-Nicolau stain, or PAP stain for short. This specific staining technique is fantastic because it highlights the cell's nucleus and its chromatin in vivid detail, making it much easier to see important features. The process involves a series of dyes and rinses that color the nucleus and cytoplasm differently. After staining, the slide is ready for the microscope. We typically start our examination at a low magnification, like 10 times, to get an overview of the slide. Then, we move up to 40 times, or even 100 times, using immersion oil for a closer look at individual cells. For more advanced research, scientists might even use fluorescence or electron microscopy to see incredibly fine details that are invisible under a standard light microscope. So, what are we looking for under the microscope? A healthy oral cell sample has a very distinct appearance. The cells are uniform, the nuclei are small and regular in shape, the cytoplasm is clear, and the chromatin inside the nucleus is evenly distributed. But when things go wrong, the signs are clear. Abnormal cells often have enlarged or irregularly shaped nuclei. A key indicator is the nucleus to cytoplasm ratio. If the nucleus looks too big for the cell, that's a red flag. You might also see cells with multiple nuclei or other unusual structures. So, the expected results fall into a few categories. We can confirm the presence of healthy cells, which is great news. We can detect early abnormal changes that might point toward cancer or precancerous conditions. Or, we can identify inflammatory or infected cells, suggesting a different kind of problem. Let's clear up some frequently asked questions. What's the main purpose of this exam? Simply put, it's for the early detection of oral diseases and cellular abnormalities. What kind of cells are we collecting? We're mostly getting superficial and intermediate cells, but basal and inflammatory cells can also be present, especially if there's an issue. How are samples collected again? Just remember the 4 key steps: rinse, scrape, transfer, and fix. Which stain is best? For detailed cytology, the Papanicolo stain is the gold standard. And how do you tell the cells apart? Healthy cells are regular and uniform, while abnormal cells are often irregular, with large, dark nuclei. Are there any challenges? Absolutely. Getting an insufficient number of cells, dealing with a lot of inflammation that obscures the view, making staining errors, or misinterpreting the results are all potential hurdles. To sum it all up, the oral cell examination is an incredibly powerful, yet simple, tool. It's vital for early diagnosis in a clinical setting, but it's also a fantastic educational experiment for students and researchers. It teaches valuable skills in sampling, microscopic preparation, staining techniques, and how to interpret cellular morphology. We hope this guide has shown you just how much you can learn from a simple cheek swab. If you found this video helpful, please give it a like, and don't forget to subscribe to our channel and hit that notification bell. That way, you won't miss out on any of our future adventures into the world of science and microscopy. Thanks for watching.